A very good evening to everyone joining us for what promises to be an enlightening session. I'm Prachi Sharma and it's my pleasure to be a host for today's session discussion Emerging Substance Trends and its Analysis. Forensic science is a dynamic field that demands constant attention. As now substances emerge, our analytical tool and expertise must evolve just as quickly. Today, we focus on the front line of this challenge, exploring the latest trends and sophisticated techniques required to address them. To lead us through this complex and fascinating topic, we are honored to have an academic leader and expert in the field, Ms. Apoorva Narad, ma'am. Ma'am is the HOD and Assistant Professor in the Department of Forensic Science at Bhoomi Uttarakhand University. Her qualification and professional contribution highlight her deep domain knowledge. She holds Master of Science in Toxicology from NFSU and has qualified UGC NET PhD. She is an active program coordinator in her department, managing academics, experimental learning, and departmental collaborations. Her expertise isn't limited to the classroom. She has presented several papers and posters on critical public health issues, including the toxicity in microwave safe food containers and the impact of phthalates in cosmetics and sanitary products. Her research expertise is evident through her publications, including an expected review article in Scopus Index Journal on the toxicity in microwave safe food containers. In short, we have with us a dynamic assistant professor, a committed researcher, and a certified young role model ready to share her insights. As a reminder to our audience, please note down your questions in the chat box and we will address them following the main presentation. Now, without further delay, let us warmly welcome ma'am to enlightening us on our session. Ma'am, this session is all yours.
Yeah. Am am I audible now? Hello.
Yes, ma'am.
Okay. So good evening, everyone. This is Apoorva Narad, and thank you, Prachi, for such a wonderful introduction. Hello. Hello.
Ma'am, you are audible properly. You can start.
Hello. Hello.
Yes, ma'am.
Can you hear me now?
Yes, ma'am, you are audible.
Okay. Thank you. Thank you, Prachi, for such a wonderful introduction. And without further ado, also before starting, I'm really sorry for the delay that happened unexpectedly. And now, without wasting your time, let's move forward towards today's topic. Okay. So, our today's topic is the emerging substance trends and their analysis in toxicology. Many a times you might have already attended lectures on NDPS and what all common substances are covered under the NDPS Act and how are they analyzed in forensic science. However, as the time is passing by, the trends in the market are changing, and so are the compositions of the products, right? And with each changing trend, we need to change our analytical techniques and also the way that we sample our evidences and how do we analyze it. So here we'll be understanding what all new psychoactive substances are there in the market and what are the analytical challenges that we face. So moving further, let me quickly give you an introduction as to what are the new psychoactive substances and why is it important to proceed with the evolving perspectives of the psychoactive substances. So new psychoactive substances are basically synthetic or natural compounds which are actually composed to mimic the effects of the previously known psychoactive substances. However, because they evade our legal restrictions, they are not producing it, want to go unnoticed and untraced, and hence they change and switch on the compositions so that they can go unnoticed. So these substances are continuously modified at the molecular level, and they create a challenge for regulatory and forensic communities while detecting them. Now this is important to note that these substances, they represent a very critical health crisis where the implications of these substances on the human health is never ending. Okay, so these substances, they pose upon unexpected, unresearched challenges in their target or their host organism, and their pharmacological profiles are often unknown because those substances are not researched, and hence their potencies are also unpredictable, because of which their dosage, their lethal effects, their exposure routes are often unstudied, and hence they have effects on the body which even the producer is unaware of, and hence it can lead to accidental poisonings, overdosages, because of which loss of human life, or maybe it can lead to addiction as well. So the key concerns while we study about the psychoactive substances is that they have, as I mentioned, they have unknown potency which can lead to overdosage. Their molecular evolution is very rapid, because of which the detection gets hampered. Now identification and quantification also becomes difficult, because as of now we have a very restricted library of psychoactive substances. Now if they are getting changed in molecular level, so that will lead to again analytical difficulties, because the studies and the researches are of limited substances, hence we know the solvents or the solutions or the analytical techniques are very limited with us. And because of that, the references that we use in our examinations are also less in number. Apart from that, since I told you that they are more toxic as compared to the previously known compounds, their clinical data is also very less. So all in all, what I want to say is ki, for example, you are well aware of cannabis, opioids, then you have amphetamine, barbiturates. Now for um, example, there is a producer who mixes all the properties of these compounds, prepares a new drug in the market, and they sell it ki isse bahut acha nasha hoga, or it is a very good intoxicant and you can use it. Just keep it under your tongue, just take it with the powder, snort it up, eat it up, smoke it up, and then the person consumes it and he himself does not know what are the clinical implications of that. And that is why global surveillance systems and reporting of these increases of the NPS substances are very important. So now what are the emerging trends from 2023 to 25? Let's see. So recent surveillance data from international and national agencies reveal the patterns in the emergence and distributions of novel psychoactive substances. The toxicological community faces accelerating challenges as the diversity and potency of these compounds continue to increase. Now, here I have mentioned 4 different categories of such compounds. Firstly, we have synthetic opioids, then we have xylazine adulteration, then we have synthetic cannabinoids, and then we have designer stimulants. Now, synthetic opioids, as of now, we were aware of fentanyl and nitazines, right? But what is happening now that their potencies have been increased up to 1,000 times as compared to morphine, which is a natural opioid, right? Because their compositions are getting changed. For example, if the natural morphine is acting at 100 micrograms, maybe these synthetic opioids might act as less as 5 micrograms, picograms, depending upon the compositions of the compound and whatever is being marketed. Then we have synthetic cannabinoids. Now, cannabinoid again is a natural psychoactive substance. If I talk about when we add the word synthetic in front of it, it simply means where there is man made interruption or interference with that drug. So, fourth generation synthetic cannabinoids continue to circulate in the market with compounds like MDMA, 4N Pinaka, which shows extreme receptor affinity. WHO reports over 200 synthetic cannabinoid variants identified since 2020. So we are not even 5 years down the lane, and there are over 200 cannabinoids in the market which are being circulated among the people who consume drugs. So I guess these statistical data is very much enough to impress on the importance of this topic as to why do we actually need to study this. Then we have xylazine adulteration. So the veterinary sedative xylazine, or you can say is as trank, is increasingly found in illicit opioid supplies across North America and Europe, complicating the overdose presentations with severe dermatological complications. And we have naloxone resistant sedation also. So as all of you know that tranquilizers are generally tranquilizer cases are generally given the antidote as naloxone. So if imagine the seriousness of the compound where the drug itself is antidote resistant, the drug is not being treated because like with the help of the antidote, right? So that is how dangerous these compounds are. Coming to designer stimulants, synthetic cations and novel stimulants persist in recreational drug markets. And Indian NCB, which is the National Narcotic Control Bureau, seizures show sustained circulation of these stimulants, particularly at rave venues and through darknet distribution channels. So in India, if we talk about our country, these compounds have seen a rise in the distribution through darknet channels and as well as the rave parties that happen mostly in the venues like mountains. Then we have beaches such as Goa, then mountains like Kasol. So these are generally known as the rave capitals of India, and these places have mostly seen the increase in these in the distribution of these compounds. Coming to a list of the emerging drug classes. So, if I talk about synthetic opioids, we have nitazines, where you have isotone nitazines, metonitazines, fentanyl analogs, which are carfentanil and acetylfentanyl. So, these effects that you see in the body, we have potent micro-opioid receptor agonism, rapid onset analgesia. So, if I talk about agonism, I hope that you know how drugs work in our body. We have synergism, antagonism, agonism, right? So it simply means that these drugs have more affinity and they produce an accelerated effect in the body. Then the toxicological risk using synthetic opioids is that it causes profound respiratory diffusion, unconsciousness, and death at nanogram doses. All right. So I don't know if many of you know, nitazines and fentanyl were those compounds whose LD50 was somewhere ranging up to micrograms. And if I talk about the emerging compositions of these compounds, it has reached up to nanogram doses. Okay. Then we have adulterants such as xylazine or trank. This is also known as zombie drug. Now, why is it known as a zombie drug? Because of its effects that it poses in the body. Okay. So, as you might be knowing, that zombies are generally organisms or maybe people, I would say, who are dead. Their skin is dead, there is no receptivity on the skin. So that is what these adulterants cause you. Anyone who is consuming it or is under the overdosage of these adulterants, they can suffer from skin necrosis, non-opioid sedation, bradycardia, And then you have agranulocytosis, wherein the agranulocytes in your body, they start dying. So the primary effect that it causes is alpha 2 androgenic agonism, deep sedation, and analgesia. Then we have synthetic cannabinoids such as MD, MB4N Pinaca, 5F ADB, ADB Butinaca. Now these are basically the chemical names or the scientific names of synthetic cannabinoids. So it causes acute psychosis, seizures, cardiotoxicity, and kidney injury as well. Coming to, we have synthetic cathinoids where we have N-ethylpentolene, 3-MMC, mephedrone, which is also known as bath salts, and drone. So these compounds generally cause dopamine inhibition, norepinephrine inhibition in your body, and it stimulates your body. So it can cause, because it is stimulating your body, it can cause severe agitation, hypothermia, cardiovascular collapses, serotonin syndromes also. Then we have designer benzodiazepines. For example, we have flualprazolam, then you have etizolam, and then you have bromozolam, which is also known as designer benzos. So this works on the GABA receptors that you have in your body, and it can also cause anxiolysis. Toxicological effects of designer benzos or the benzodiazepines lead to CNS depression. It can, sedate your central nervous system, can cause severe amnesia, respiratory failure in combination, or severe dependence on the drug also. Now, if you see, I've put a note here in the bottom of the mm, presentation or the slide, that each class exhibits pharmacological signatures that require targeted analytical approaches. These ultra high potency of synthetic opioids necessitates the detection limits in the picogram range, while the structural diversity of synthetic cannabinoids demands broad spectrum screening capabilities, meaning that till now, when we were using the traditional compositions, the levels were till micrograms or till milligrams. However, these synthetic compounds can even range to picogram range, which is not only hampering our analytical ranges, but it's also leading to screening incapabilities also. Coming to the pharmacological and toxicological features and the mechanism of action of these drugs. So emerging substances, they exploit characterized neuropharmacological targets through novel molecular scaffolds. Synthetic opioids demonstrate extreme opioid receptor affinity with prolonged dissociation kinetics. Xylazine acts as a central alpha 2 androgenic agonist, which is opposite of synergism. Then we have producing sedation independent of opioid pathways, synthetic cannabinoids that function as full CB1 receptor agonist, unlike partial agonism, which is seen in delta 9 THC, which is cannabinoids' natural composition, explaining their disproportionate psychotomimetic effects, which means that they mimic the effects of these natural compounds in your body. So the toxic manifestations are that in the central nervous system, you can see altered consciousness, seizures, psychosis, and coma. Respiratory manifestations are that it can lead to severe depression of the respiratory system, apnea, hypoxia, which leads to anoxic brain injury. Cardiovascular manifestations include arrhythmias, hypertension, myocardial infarction, sudden cardiac death also can take place because of these manifestations in the body. So, what happens on like how do these drugs function in your body? So basically, these psychoactive substances enter your body through inhalation, injection, ingestion, or transdermal absorption, which are the normal traditional routes of exposure of any drug. Then it binds in with the receptors in your body. Now it can lead to agonistic behavior and antagonistic behavior, meaning it can either increase the receptivity or it can decrease the receptivity in your body. Then you have toxic effects, which is organ system dysfunction, CNS depressions, respiratory failures, and so on. Coming to polydrug interactions and clinical complexities of these drugs, so the contemporary overdose landscape is characterized by polypharmacy toxicity. The co-administration of synthetic opioids can lead to complete resisting sedation in the body, which will obviously which will obviously lead to increasing dosage in the body, because your body conclusively becomes resistant to these drugs. Concurrent benzodiazepine use potentiates respiratory depression. Synthetic stimulants combined with opioids produce unpredictable cardiovascular effects. This pharmacological complexity demands comprehensive toxicological screening rather than targeted single analyte testing, and necessitates sophisticated interpretation that considers synergistic and antagonistic interactions, as I mentioned earlier. Now, what are the challenges that we as toxicologists are facing while detecting these compounds? So, the analytical identification of these substances represent one of the most formidable challenges in modern forensic toxicology. These compounds are deliberately designed to evade detection, creating a continuous arm race between synthetic chemists and analytical laboratories. So, because of this, we need rapid chemical modifications. Now, clandestine basically is a name given to the laboratories where these productions take place, where they keep on changing the compositions of natural and previously occurring drugs. So, clandestine chemists systematically modify molecular structures to circumvent legal schedules and immunoassay detection. Minor substitutions are made, such as fluorine atoms or alkyl chain extensions can be done to create legally distinct compounds with similar pharmacological effects. This molecular diversity renders traditional antibody based screening ineffective, as immunoassays lack cross reactivity with novel analogs. Now, if आप लोग को पता हो तो हमारी जो एनडीपीएस की रूल बुक है ना, इट covers only limited compounds, right? जिनको वो illegal बोलते हैं. So what these chemists do is they change the composition, because of which it obviously it is deleted. It does not come under that library of the compounds that are mentioned in NDPS, right? And all the immunoassay करते हैं, हमारे पास उन्हीं की डिटेक्शन किट्स हैं. अगर कोई नया ही ड्रग आ जाएगा तो ऑब्वियसली क्रॉस लिंकिंग इम्यूनोएसिड्स हमारे पास अभी भी नहीं है, बिकॉज ऑफ विच दे गो अनडिटेक्टेड. Absence of reference standards. Distinctive identification requires comparison against authenticated reference materials. However, the lag time between substance emergence and commercial standard availability can span months to years. मतलब मार्केट में आने से लेकर हमारे पास उन्हें आइडेंटिफाई होने तक इट कैन टेक मंथ्स टू इयर्स. तो आप सोचो, तब तक तो वो ऑलरेडी मार्केट में इतना ज्यादा सर्कुलेट हो चुका है, and it is going unnoticed and undetected as well. So laboratories are forced to rely on predicted mass spectra, fragmentation patterns and retention time estimations, increasing uncertainty in identification. तो अब हम करते क्या है उस केस में कि जहां हम सिर्फ पीक्स स्टडी किया करते थे, उसकी जगह अब हम फ्रेगमेंटेशन पैटर्न्स भी स्टडी कर रहे हैं कि क्या हमें इसमें कोई और कंपाउंड्स मिल रहे हैं जिनके फ्रैगमेंट्स हमें मिल रहे हैं. रिटेंशन टाइम वेरी हो रहा है कि नहीं. फॉर एग्जांपल मॉर्फिन का रिटेंशन टाइम एक्स है, और अभी जो हमारे पास कंपाउंड आया है उसमें मॉर्फिन का सब्स्टिट्यूट तो है, बट उसका रिटेंशन टाइम उससे इधर उधर है. देन वी कैन से दैट यस, द कंपाउंड इज मिक्स्ड विद समथिंग एल्स आल्सो. Ultra low detection thresholds. So the extraordinary potency of compounds like carfentanil or isonitazone necessitates the analytical methods with picogram sensitivities. Traditional instrumentations may lack sufficient detection limits, requiring high-resolution mass spectrometry and optimized sample preparations to concentrate analytes while removing interfering matrix components. Coming to matrix complexity and postmortem artifacts, biological matrices contain thousands of endogenous compounds that produce ion suppression and interfere with chromatographic separation. Postmortem samples introduce additional complexity through redistribution phenomena where drugs migrate from tissues into blood and enzymatic bacterial degradation that can destroy target analytes or generate artifactual compounds. Now, आप ये समझो कि अभी तक हम एनालाइज नहीं कर पा रहे हैं थ्रू इंस्ट्रूमेंट्स कि इसमें कौन सा कंपाउंड है या क्या कॉम्पोजिशन यूज हुई है. तो आप सोचो पोस्टमार्टम जो मैनुअली कर रहा है उसको वो क्लिनिकल इम्प्लीकेशंस पढ़ने के लिए कितना चैलेंजिंग होगा कि हर बार दैट द पोस्टमार्टम इज बीइंग डन अ न्यू कंपाउंड इज बीइंग एक्सट्रैक्टेड फ्रॉम द बॉडी और न्यू clinical implications are seen. These challenges necessitate a paradigm shift from targeted analysis towards broad spectrum, untargeted screening using high resolution accurate mass spectrometry, coupled with comprehensive toxicological databases and sophisticated data mining algorithms. Now let's see how is the sample collection and preparation done in these cases. So rigorous sample collection and preparation protocols are the foundation of reliable toxicological analysis. The quality of these results is directly dependent on the proper specimen handling, preservation and extraction methodologies. तो सबसे पहले तो हमें सैंपल सिलेक्ट करना है कि हमें सैंपल चाहिए कौन सा, राइट? तो इफ यू कैन सी ओवर हियर, first, blood from femoral or subclavian bone is taken. Then urine is one such biological fluid that shows the longest detection window. So इसलिए हमें urine collect करना भी उतना ही important है. Vitreous humor, which is always protected from contamination in the body, and it reflects the antemortem levels of the drug itself, so that is also important. Then we have liver tissue, which shows the highest drug concentration because वहीं पे blood purify हो रहा है. Useful when the blood is unavailable. So इसीलिए हम kidney tissues को तब use liver tissues को तब use करेंगे तब हमारे पास blood unavailable है. And hair, which provides chronic exposure history. जब वीक से लेके मंथ्स तक में अगर वो ड्रग यूज किया जा रहा है तो हम हेयर को यूज करेंगे क्योंकि उसकी विंडो जो क्रॉनिक एक्सपोजर की होती है वो सबसे ज्यादा होती है. Coming to preservation, so samples are refrigerated from 2 to 8 degrees or they are even frozen, ranging from -20 to -80 degrees Celsius for long-term storages. And sodium fluoride is used as a preservative to inhibit enzymatic and bacterial metabolism in those samples. Then anticoagulants such as EDTA, sodium fluoride, potassium oxalates are used to prevent the coagulation of the blood. Apart from that, pH stability should also be maintained so that hydrolysis or ester hydrolysis can be prevented in the drugs that contain esters in them. Now for extraction methods, protein precipitation, centrifugation, liquid-liquid extraction and solid phase extraction can be used. I am expecting that you are aware of these techniques where acetonitrile or methanol is used to precipitate out proteins in the protein precipitation from blood. Centrifugation, all of you are aware, where we use centrifugal force to separate the components from the matrix. Liquid-liquid extraction is used to separate the organic solvents from the lipophilic compounds. Then solid phase extraction is used when you have limited amount of the solvent or the analyte in your matrix. And then we come to enzymatic hydrolysis, which is the last phase 2 metabolism of the drugs in your body. So many drugs undergo phase 2 metabolism that forms glucuronides or sulfonate conjugates inside the body itself. So we need to treat that evidence with beta glucuronidase or aryl sulfatase so that it can liberate the parent drug from the body, and then phase 1 metabolites can increase the detection sensitivity in our entire matrix. Reagents that are commonly used are acetonitrile, which is LCMS grade for LCMS testing. Formic acid, 0.1% is used in mobile phases. Beta glucuronidase is extracted from E. coli. And then we have internal standards which depend upon various drugs. And then we have ammonium formate buffers which are used to maintain the pH of our samples. Coming to the different analytical techniques, first we use the screening techniques where we have immunoassays and LCMS screening. Immunoassays such as ELISA or automated immunoassay platforms where we can process dozens to hundreds of samples every day are used, but they are suffering poor specificity because of cross-linking immunoassay or cross-linking antibody reactions in our body. So it can also yield false negatives in the case of emerging substances. Then we have rapid LCMS screening, which utilizes chromatographic gradients with targeted or suspect screening databases. This approach improves the specificity of the detection, and also it maintains, the quantity of the drug that is found, or it can also detect hundreds of compounds in one injection. So you see that since it is detecting hundreds of compounds, the sensitivity of the device is increasing, the sensitivity of the experiment is increasing. Now this is the confirmatory testing. Here I've compared three techniques. We have LCMSMS, LCHRMS, and GCMS. So advantages of LCMS is that the quantitative gold standard is used in LCMSMS because its sensitivity is excellent. Like its detection limits are up to picogram per ml of body fluid, right? Limitation, however, is that it requires known targets, and because of that, you cannot function with constantly changing drugs every day. So the library needs constant updation as well. So the best application is where you have to target quantification of psychoactive substances for regulatory or framework cases, right? When we have to frame regulations on the psychoactive substances, for that cases, LCMSMS is the perfect solution for that. Then you have LCHRMS, which has untargeted capabilities, so it can scan the entire component or the compound for you, and its detection limit is that it is accurate when the compound is even less than 5 ppm in your solution. However, its data interpretation is very complex. It is a very expensive instrument and it requires expertise, so not everyone can use LCHRMS. Also, it is best used when you have to discover new compounds or unknown substances in your matrix. And then we have your traditional GCMS, which is an extensive spectral library, where the library, if you might have known about NIST. So it is excellent for volatile and robust fragmenting compounds. Now its limitation is again that जो भी कंपाउंड्स वॉलेटाइल नहीं है या थर्मली स्टेबल नहीं है, आप उनके लिए इसे यूज नहीं कर सकते. एंड जस्ट इन केस इफ यू यूज इट for non-involatile or such compounds, your sample can get destroyed, okay, because it's a destructive technique. Volatile drugs, stimulants, and cannabinoids can be used and detected using GCMS. Coming to the interpretation and reporting of the compounds, so I have mentioned it in 5 different points. First one is that you need to identify the parent drug and metabolites. So detection is very important because हमें parent compound तो पहचानना है, but वो किन compositions में लाकर उन्हें नया drug बनाया जा रहा है, वो हमें evaluate करना ज्यादा जरूरी है. Because अगर हम वो नहीं कर पाएंगे तो हम नया regulatory framework develop नहीं कर पाएंगे. ठीक है. देन कोरिलेट विद केस हिस्ट्री, कि अगर पुरानी कोई केस हिस्ट्री अनरिजॉल्व चली आ रही है तो उसको आप विटनेसिंग में हमें ये चीजें पता चली, प्रिस्क्रिप्शन ड्रग है, तो इनकंसिस्टेंसीज जो है then you have assessing clinical and autopsy findings. इंटरप्रिटेशन एंड रिपोर्टिंग इसलिए भी जरूरी हो जाती है क्योंकि अगर हमें किसी ऑटोप्सी में या किसी क्लिनिकल फाइंडिंग्स में कुछ ऐसा मिला है जो अभी तक रिपोर्ट नहीं हुआ है ना, हमारी एनडीपीएस पूरी टीम को या एनसीबी को, तो उनको बताकर उसमें क्या क्या चेंजेस आ रहे हैं या क्या क्या पोस्टमार्टम फाइंडिंग्स है उनको कोरिलेट करना भी जरूरी है। क्योंकि ड्रग तो हमने पता कर लिया कि चलो फेंटेनिल है, बट फेंटेनिल बॉडी में कर क्या रहा है वो भी तो बताना जरूरी है। इट इज इक्वली इंपॉर्टेंट, राइट? एंड देन वी हैव अप्लाई क्वांटिटेटिव कॉन्टेक्स्ट। अब अगेन पता चल गया ड्रग चेंज्ड है, कॉम्पोजिशन चेंज्ड है, ये क्लिनिकल फाइंडिंग्स है, बट अगर हमें ये नहीं पता कि उसमें एक्स वाई जी कंपाउंड्स किन कॉम्पोजिशन में है, how are you going to put a label on that particular drug? How are you going to name that drug? So that is also important while interpreting or studying psychoactive substances. And then we have addressing analytical limitations कि भाई यहाँ तक तो हमने आइडेंटिफाई कर लिया, what next? How are we going to identify the compounds जिनकी हमारे पास लाइब्रेरी ही नहीं है, जिनका हमारे पास कोई कॉम्पोजिशन कोई हिस्ट्री नहीं है, ठीक है? सो एक्सप्लिसिटली स्टेट एनी लिमिटेशंस, पोस्टमार्टम रीडिस्ट्रीब्यूशन इफेक्ट्स, पोटेंशियल एनालाइट इंस्टेबिलिटी, एब्सेंस ऑफ रेफरेंस स्टैंडर्ड, सेमी क्वांटिटेटिव रिजल्ट्स। अब ये चीजें क्या है, ये हमें प्रीवियस ड्रग्स के लिए पता है, बट नए ड्रग्स की कोई हिस्ट्री नहीं है हमारे पास, तो वो लाइब्रेरी बनाना टॉक्सिकोलॉजिस्ट का काम है, राइट? और वो हम एलसीएमएस एंड एलसीएचआरएमएस से कर सकते हैं। कमिंग टू क्रिटिकल इंटरप्रेटिव कंसिडरेशंस, 3 चीजें हैं, ठीक है, मैंने मेंशन करी है यहाँ। पोस्टमार्टम रीडिस्ट्रीब्यूशन, टॉलरेंस एंड डिपेंडेंस, एंड पॉलीड्रग सिनर्जी। तो पोस्टमार्टम रीडिस्ट्रीब्यूशन से मतलब है कि कोई भी अगर ड्रग बॉडी में पोस्टमार्टम के बाद रीडिस्ट्रीब्यूट होकर फेज 1 फेज 2 मेटाबॉलिज्म्स या पैसिव डिफ्यूजन करते हुए कहीं और रीडिस्ट्रीब्यूट हो जा रहा है, ठीक है, और वो आपके टेस्ट एविडेंसेस में पता नहीं लग रहा है, का मतलब ये नहीं है कि वो बॉडी में प्रेजेंट नहीं है, तो उस ड्रग का रीडिस्ट्रीब्यूशन भी पढ़ना हमारे लिए उतना ही जरूरी है, ठीक है? टॉलरेंस एंड डिपेंडेंस, tolerate concentrations that would be in naive individuals, and hence clinical clues history are essential for interpretation. अब फॉर एग्जाम्पल कैनाबिनॉइड की LD50 या ED50 हम सबको पता है, ठीक है, तो इंसान उस हिसाब से डोजेज लेता है कि ओवर डोजेज ना हो जाए। अब फॉर एग्जाम्पल उसको कोई इंसान नया ड्रग लाकर देता है जिसकी LD50 उसे नहीं पता है, एक्सपेरिमेंटल स्टेज में तो होते हैं ये ड्रग्स, तो ना उसको टॉलरेंस पता है कि उसकी कितनी है बॉडी में, ना उसको डिपेंडेंस पता है कि किस टाइम पर आकर मैं उस ड्रग पर डिपेंडेंट हो जाऊंगा, मुझे तो ये भी नहीं पता। तो ये दिस इज वन मोर कंसिडरेशन कि जब आप अपना कोई नया ड्रग एनकाउंटर कर रहे हो तो आपको ये सारी चीजें उस ड्रग के बारे में पता लगानी ही लगानी है, फॉर अवेयरनेस पर्पस ऑब्वियसली। पॉलीड्रग सिनर्जी इज दैट 2-3 ड्रग्स मिलकर क्या कोई सिनर्जिस्टिक टॉक्सिसिटी कॉज कर रहे हैं, क्या वो बॉडी में कुछ नया कॉज कर रहे हैं, ठीक है? सो दैट इज ऑल्सो इम्पॉर्टेंट वाइल अंडरस्टैंडिंग कि इस ड्रग ने क्या चेंजेस लाया बॉडी में। फॉर एग्जाम्पल हियर आई हैव रिटन कंबाइंड रेस्पिरेटरी डिप्रेसेंट सच एज ओपियोइड्स, बेंजोडाइजेपिन एंड अल्कोहल प्रोड्यूस सिनर्जिस्टिक टॉक्सिसिटी इन द बॉडी। तो अगर कोई बंदा है या कोई एक्स पर्सन है जिसने ये तीनों ड्रग्स एक साथ ली हैं, तो कंसेक्युटिवली उस दैट इंडिविजुअल इज बाउंड टू डाई, क्योंकि उसको सिनर्जिस्टिक इफेक्ट्स के बारे में पता ही नहीं है। So that awareness should also be there, and people like us, like toxicologists, should work in this direction. So at this given point, I call for any collaborative work from any of you. If you want to work on the synergistic, antagonistic, or any as such clinical implications of these drugs in combinations, please reach out to me. You can reach out through mail or even call. I'll provide that in the group after the lecture. So here are these key takeaways: that there is a persisting challenge in emerging psychoactive substances that represent continuously evolving threat to public health and a challenge for forensic toxicology. The synthetic drug marketplace adapts faster than regulatory and analytical responses, necessitating the agile, forward thinking approaches to detection and identification. Analytical innovation. So, the next generation analytical platforms that include high resolution mass spectrometry, ion mobility separation, and artificial intelligence driven spectral interpretations promise for comprehensive untargeted screening where the compounds are going undetected. Collaborative imperative, where the effective response requires collaboration among toxicologists, clinicians, law enforcement agencies, and international monitoring networks. So, the takeaways from this lecture for you all is that synthetic opioids, particularly nitazines and fentanyl analogs, pose the most significant mortality risk. Adulterants like xylazine complicate the overdosage representations in the body. हाई रेजोल्यूशन मास स्पेक्ट्रोमेट्री इज एसेंशियल फॉर अनटारगेटेड डिटेक्शन वेयर आपको पता नहीं है कि आप क्या ढूंढ रहे हो बट आप ढूंढ रहे हो कुछ. Proper sample collection, preservation, and preparation is critical for reliable results. Interpretation requires integration of analytical data with clinical and investigative context, and continuous education and method validation are necessary to keep pace with emerging threats. So, for today, I am putting an end to my entire session by saying that toxicologist's role has evolved from simple detection to forensic detective work, requiring sophisticated analytical tools that comprehensive databases and expert interpretation to identify and characterize the ever changing landscape of psychoactive substances threatening to public health. And as you might have studied in the very first lecture of forensic science, that our work is to provide justice and circulate awareness in the society. So, our work gets increased by severe folds when these clandestine chemists are regularly working on introduction and discovery of new drugs. So our work becomes even more important for public health and their safety. So I'm opening the session for any questions if you have, and then let's resort to each of those questions one by one.
Wow, what an incredible, insightful session. Thank you, ma'am, for shedding light on the complexities of emerging substance trends and their analysis. Your expertise, especially your discussion on analytical techniques and the toxicology principle, was invaluable. So now we will take the questions from the audience.
Prachi, I by mistake disconnected the call. So if there are any previous questions in the chat box, can you please just copy it down for me again?
Ma'am, there aren't any question.
Okay, oh,
Participants,
all
if
right.
you have any question, please ask.
Also, I am putting down my email ID for all of you if you want to contact me for any collaborative work. I'll be more than happy to assist you all.
Does anyone have any question? If not, we will proceed with the session. Ma'am, I think there are any question.
No worries. Even if they have any queries after the lecture, they might contact. Okay, so they can contact the team whoever was taking care of the registrations. Guys, if you have any questions or any queries, you can either mail me up directly, or you can also contact the registration team so that they can provide the question to me. However, I am quickly sharing the presentation with sir so that you can refer it for future references also.
We have gained a vital understanding of the current challenges and the innovative solutions being deployed in forensic science. As we come to close to this today's session, it's time to express our heartfelt gratitude. First, a warm thank you to our speaker for generously sharing her time, profound experience, and knowledge with us. Your presence today has enriched this session and inspired all of us. And a big thank you to all participants for being such curious audience. Your active participation and thoughtfulness question are what makes session like these truly worthwhile. Once again, thank you everyone for joining us. Stay connected, stay curious, and most importantly, inspired. Thank you so much.